Hey, today we're going to learn about biotechnology. So here we are, we're going to learn about biotechnology, and we're here with Dr. Brown. So what do you have for us today?
Yes, well, biotechnology is the exploitation of biological processes for industrial and other purposes, especially the manipulation of microorganisms used for the production of antibiotic hormones. Let's go into the lab to take a closer look.
Thank you.
Now, modern uses of technology have allowed us to make advancements in biological research. Let's start with the first major subsection of biotechnology, gene cloning. Now, gene cloning is where a particular gene of interest is located on a segment of DNA and then inserted into another organism. So first what we do is we have to take our pile of DNA with all of our genes in it and locate our gene of interest. After we do that, we take our restriction enzyme. I'm not sure where it is.
It's right here.
Thank you. Now, after taking a restriction enzyme, we cut out a little piece of DNA with that gene on it. Now, after doing this, we use the same restriction enzymes to cut a piece of a plasmid. We have that cut piece right here. And after that, we combined the cut piece of the plasmid and the gene of interest together. And once we do this, this can be inserted into a bacterial cell, and the bacterial cell will reproduce.
So yeah, I think that whole concept of gene cloning is pretty interesting.
Yeah, well, this actually relates to the whole concept of gene editing. Now, gene editing is basically the insertion, deletion, or replacement of DNA at a specific site in the genome of an organism. Now, basically, this is done in a lab with engineered nucleases, known as molecular scissors.
Alright.
Now, there's a number of nucleases we can use. Some of them you might be familiar with, like CRISPR, Cas9, ZFNs, or TALENs.
Right. Okay, so I have a question. What if there's a gene that's already in the organism that I want to change how it's expressed?
Well, biotechnology can be used for gene expression. The popular way that this is done is that the antibiotic tetracycline can be used to change the expression of the CRE gene in specific cell types.
Right.
And doing this actually changes the arrangement of chromosomal DNA.
Oh, okay.
Alright, so here we are. Let's start with the practical uses of biotechnology. You can actually step outside for this demonstration.
Are you sure?
Yeah, just one second.
Like right here?
Oh yeah, yeah.
Fine here?
Yeah, it's fine. Don't worry about it. Hey, you work here at the lab now? Are you new?
Thanks.
Well, okay. Well, while you're here, you want to learn about some practical uses of biotechnology.
Yeah.
Now, here's actually a very practical use of biotechnology. So many detergents like this one and others often have enzymes that help to remove stains, which is pretty important for cleaning dishes. So, if you read that right there, does that say that this has amylase in it?
Yeah.
Yeah. Okay. Well, amylase helps to remove starch, and
Wait, does that
proteases right there?
Yeah. Wait, is that lipase then? Is that lipase?
Oh yeah. Well, first of all, proteases help to remove protein stains, but lipases help to remove fat stains, which is good for removing grease.
Okay. That's what I like to hear.
Yeah. So now, because these do contain enzymes, though, that's why these won't work that well if there's if the temperature or pH is extreme. So we just So first we just go ahead and put some of this detergent on our dirty plates here. Just give it a second. There it is. Then we go ahead and scrub it down. And then, so those will be cleaned, which is why this is an excellent use of biotechnology. Let's move on to the next one now. Oh, hey there. You want to,
hanging, bro.
oh no, yeah, sorry about that. You want to see another way that we can use biotechnology in our everyday lives? So just right over here. You see these plants?
Yep.
Well, these are actually cotton plants.
Okay.
So they've been genetically modified. Now, biotechnology can be used to modify plants by making them resistant to certain diseases or pesticides, or bugs. So basically, a gene from one of the bacilli bacteria was inserted into this cotton plant, and that gave it protection against the native bollworm and the cotton bollworm, which is why you can see that it's so beautiful, and you can actually see some of the cotton is starting to grow here. Look at that. You see that?
Oh, right, yeah, I see.
Yeah.
Okay.
So that cotton's used to make these kind of shirts, actually. So
wow.
If you didn't have that, you wouldn't be wearing a shirt right now. So
That's crazy.
yeah. All right. Well, we actually have one more practical use. So that's back inside in the lab.
All right.
Let's go.
Thank you.
So our next practical use we're going to be talking about is vaccines. So this right here is a vaccine. So vaccines are a big aspect of biotech because basically what it is that a weakened form of a virus is introduced into someone's body, which makes the body's immune system more capable of fighting off the virus when it actually comes.
Right. Like flu shots.
Right. So these usually have to take a lot of testing and procedures for that, but because of our technological advancements, this has actually become a very accessible and convenient method of fighting off disease.
Okay, so this one is it safe to use right now? Is it ready or?
This one?
Yeah.
Yeah. Well, it hasn't been tested yet, but it's safe.
Okay.
I'll be right back.
Oh, yeah.
Lipases, how fascinating.
Hey, so is it safe or
Oh,
the vaccine? Is it safe?
yeah, it's fine. It's fine. We still need some tests and stuff, but we're good.
Okay.
So our next major subsection, our last one, is going to be gene therapy. So let's go check that out. So basically, gene therapy is the transplantation of a working gene in the place of a defective gene, which can be used to treat certain disorders or certain issues with an individual. So basically the way this is done is the new gene is injected into an adenovirus, which is then introduced into the human cell. And if this procedure is successful, then the human cell should start making the protein effectively. So let's go over here and take a look. So for example, hemophilia is a genetic disorder in which there's a defective gene, and individuals with the disorder cannot properly clot their blood, which results in severe bleeding and can be life-threatening. So you can actually see this patient right here has hemophilia and got a paper cut.
Oh wow.
So actually, one way that this can be treated, as several trials and clinics have done, is the therapeutic gene will be inserted into the liver of the affected individual, which allows them to clot blood at the proper time.
Oh, okay.
So I'm actually going to go ahead and demonstrate that for you.
Alright.
Hey,
What happened?
you okay?
Yeah.
Okay, cool. Bye. So yeah, that's all there is to it. Really, that's how gene therapy works. But there's still different disorders that need gene therapy, while some don't. There's still a lot of research being put into it. It's not 100%.
Well, I mean, I thank you. I actually learned quite a bit. So.
Yeah, of course. Well, I'm glad we could meet and you could learn about biotechnology. So
Yeah.
yeah.
Okay, well, have a good day.
Yeah, you too.